going to use this PowerPoint to go through the phases of mitosis. Remember that mitosis occurs in somatic cells. It is used for cell replacement, cell growth, and it's one part of the overall cell cycle. So if we look at the entire cell cycle, we can divide it into 3 phases. We have interphase. The cell spends most of its time in interphase. This is when it's undergoing normal cell activities. Mitosis particularly is the time of nuclear division. The DNA is going to split and divide, and then cytokinesis occurs after mitosis. Sometimes there's a little bit of overlap between the end of mitosis and the beginning of cytokinesis, and once the nucleus divides, then the cytoplasm divides. So let's start by looking at interphase. As I said, it's the longest part of the cell cycle. It can take up to about 80 or 90% of the cell cycle. And during interphase, the cell is just undergoing normal processes. So it's making proteins, it's making lipids, it's making carbohydrates, it's growing bigger. There's nothing unusual going on during interphase. Interphase is divided into 3 stages. The first one is called G1 or gap 1. During G1, the cell grows and functions normally. So all those processes I just told you about. S phase, S stands for synthesis, and this is when the DNA is copied. And we learned already about DNA replication and how that occurs. So replication of the DNA occurs during S phase of interphase. S phase is followed by G2. During G2, the cell is preparing for division. So for example, it's making some extra proteins that are going to attach to the chromosomes and move them around the cell during mitosis. Okay, here's a picture at the top that sort of shows you a timeline of the whole process. So you can see mitosis is a very short portion of the entire cell cycle. Then G1, S, and G2 take up most of the cell cycle with interphase. So we're going to talk about mitosis in 4 different stages. Understand that there is no sort of break or separation. It is a simultaneous process, but it's easier for us to divide the events into 4 different parts: prophase, metaphase, anaphase, and telophase. So a lot of times you hear people say PMAT to remember the order of the phases, and then that takes the first letter of prophase, metaphase, anaphase, and telophase. So let's start with prophase. Okay, in prophase, the nucleus starts to break down. So the nuclear envelope starts to disintegrate. The chromosomes also condense. Remember, chromosomes are made up of DNA wrapped around histone proteins, and during mitosis, the cell wants to make those chromosomes very compact. It's going to be easier to sort of move them around the cell if they're smaller, and there's less chance of them getting tangled, and there's more chance of mitosis occurring properly. Okay? The spindle also forms. So what is the spindle? The spindle is made out of microtubules, which is a cytoskeletal protein that we learned about earlier this semester. So the cell makes lots and lots of microtubules to form the spindle apparatus. And that's what's happening over here. In animal cells, the spindle apparatus comes from a structure called the centriole, and they radiate out of the centrioles. And so there's going to be spindle apparatus at either end of the cell. The microtubules are going to come towards the center of the cell. By the end of prophase, the nucleus is completely gone. The nuclear envelope has disintegrated, and the spindle apparatus is completely formed, and the chromosomes are really condensed. And that's why we can see them here through this microscope picture. These are all of the chromosomes, and you can sort of see some of the spindles. Here in the cartoon, we can easily make out that the spindle has attached to the chromosomes. You can see the chromosomes are duplicated as well. So replication occurred during interphase, and the duplicated chromosomes stay attached, forming these sister chromatids. So a sister chromatid is 2 chromosomes attached to one another. You can see that the spindle's coming from opposite ends or opposite poles of the cell, and the purpose of the spindle is going to be to move and manipulate the chromosomes through the following stages of mitosis. So think of prophase as sort of setting up the entire process, getting rid of the nucleus, forming the spindle, attaching to the chromosomes, so now we can move them around and separate them. The next stage is metaphase, and when I think of metaphase, I think of midline, because that's what's going to happen to the chromosomes. The microtubules of the spindle are going to pull and push those chromosomes until they are all lined up in a row right on the middle of the cell. Okay? So this arrangement, again, is due to the spindle apparatus. The middle of the cell is sometimes called the spindle equator, so just like the equator of the Earth, sometimes it's also called the metaphase plate. So you can use either one of those terms interchangeably. They are both acceptable. So metaphase chromosomes line up on the midline. Anaphase, they split. So the sister chromatids are going to split at the centromere, and one chromatid is going to get pulled to either end of the cell. Again, this is due to the spindle. Those microtubules are pulling and pulling and pulling from the opposite ends, and finally those sister chromatids split. This is what we want to happen, because right now the cell has too much DNA, right? It only needs to have half of that number of DNA. It doesn't need to have a replicated chromosome, so we're going to pull it apart. That's what happens in anaphase. In telophase, the chromosomes reach either end of the cell, and the spindle has done its job, so it breaks down. The cell no longer needs all of those microtubules. The nucleus reforms. Okay, so you can see that there's two nuclei that are going to reform around both sets of chromosomes. And the chromosomes begin to uncoil. They're not as tightly wound around the histone proteins anymore because they have reached their final destination. You can also see that the cell is starting to divide through cytokinesis, and I mentioned earlier that sometimes there's an overlap between telophase and cytokinesis. Here in this microscope picture, you can see that there are two new nuclei in this one plant cell. So those are the stages of mitosis. So now let's talk about cytokinesis. Cytokinesis is specifically the division of the cytoplasm, and it's different if we're talking about animals versus plants, because what do plant cells have? The cell wall. Okay? So plant cells is a little bit more complicated because we have to take into account that cell wall. The process in animal cells is called cleavage furrow, and in plant cells it's called cell plate formation. Cleavage furrow is basically the cell pinching into, and it's due to actin filaments. Actin's another structural protein, part of the cytoskeleton, and it basically takes the plasma membrane and makes it pinch inwards at the top and at the bottom, and that's what this picture is showing you here, until it pinches and it meets in the middle, and then one cell is split into two. Okay, so here you can see that that pinching occurs along the former spindle equator or metaphase plate, and it's a very easy, straightforward way to create two new daughter cells. Plant cells, however, cannot pinch because of that cell wall, so they go through a process called cell plate formation. Little vesicles, little packages of cell wall forming material like cellulose and other proteins and carbohydrates sort of migrate to that former spindle equator and line up, and as more and more vesicles come and join, they start to join together until eventually there's so many vesicles that a new cell wall is built right along the middle of the cell, and that one original cell splits into two. Again, this cell can't pinch because it has the cell wall, so again it goes through this cell plate formation. So by the end of mitosis and cytokinesis, we have two new daughter cells. They are identical. They have the same chromosomes as the parent cell, and the chromosomes are back into an unduplicated form. As soon as cytokinesis is complete, these cells enter back into interphase and start undergoing regular cell activities.